I am Nia Indelicato. I am the Assistant Director of Programs at TRI, and I just want to welcome everyone to the kickoff for this season's Research Fundamentals Seminar Series. We've been on a bit of a break with COVID, but now we are back, and even though we're meeting virtually, we are glad to have you here. Our speaker today is Dr. Virginia Miller. She's from the Mayo Clinic, although I believe she is retired now, so she is currently formerly of the Mayo Clinic, where she was the director, I believe, of the Women's Health Center there. She is a professor in surgery and physiology, and she's going to speak to us today on using sex as a biological variable in research. We are very excited to have her here. I will turn it over to Dr. Miller. Thank you, guys.
Well, thanks very much for inviting me. I'm very disappointed that I can't meet with all of you in person. I was really looking forward to coming down there in March. And of course, that was right after we were all isolated. And so I hope everybody's opening up now, and it's exciting. Yes, I've officially retired June 30th, but my mind hasn't turned to mush yet. And the concepts that I'm going to present to you today, I still think are very relevant to what you're going to do moving forward. So I guess I'll share my screen and see if we can't get started with this. So let's talk about sex as a biological variable, especially how it relates to research. Okay, I'm sharing you my funding sources because if there's CME credit here, this needs to be declared. I don't have any financial interests. And what I'd like to do in the short time I have with you today is really articulate why we need, why there's a need for sex-based research, articulate the NIH policy about how to, requiring sex as a biological variable to be included in research grants, differentiate sex from gender, and really identify key components of a research strategy, hopefully that'll be able to have your proposals developed a little more clearly in addressing sex as a biological variable and make them more successful. So first of all, why do we need to consider sex as a biological variable in research and clinical practice? Well, research is really the driving edge for education and clinical practice. And when we consider a sex-based approach, it really is the essence of a holistic approach to patient-centered healthcare. Now, of course, I'm at Mayo Clinic, so we are always talking about the needs of the patient come first and centered healthcare. But when you think about NIH in your applications going forward, really the purpose of NIH was to improve the health of the nation. So that requires basic understanding of systems in order to apply that to the health of the nation. So although it may be far-fetched for some of you, or seemingly so, if you're doing some very basic mechanistic work, you need to start to reframe your ideas in terms of what the ultimate outcome of those observations might be. So in 2014, it seems just like yesterday, but when you think about it, it was 6 years ago that Francis Collins said it was crucial for experimental design that are all too frequently key components are ignored, and one of those is sex difference. Considering sex does improve transparency of your data and reproducibility. So NIH expects researchers to study both male and female vertebrate animals and humans. Now the key thing here is we're applicable, and we'll talk about that, but this expectation is gaining momentum within study sections. It was so slow to get started, but now it's really at the forefront. So let's start. What's the difference between sex and gender? Well, the Institute of Medicine has defined sex as being male or female according to reproductive organs and functions assigned by the chromosomal complement. This was a definition that was put forward in 2001, the Sex Matter, their hallmark publication. And I think it's really important to emphasize this now because there's a lot of discussion out there that this is not an appropriate definition and a lot of confusion. And I think we really need to get back to the biology and really understand why this definition was created as it was. I'd like to remind everybody that the human karyotype does have 22 sets of chromosomes, but a set of sex chromosomes, XX for female and XY for males. this karyotype is in every nucleated cell in the human body. Therefore, one of the conclusions from that hallmark publication of the Institute of Medicine was that every cell has a sex. At the genetic level, all sex differences result from the imbalance of the X and Y chromosome. These are the non-gonadal effects and are considered sex chromosome effects. You can't get away from this. And these are present at the very earliest stages of development, and even start to show up as differences before the development of the reproductive organs, gonads, and hormones. But you have to keep, so let's think about these chromosomes a little bit. The X chromosome is pretty big, has a lot of genes on it, and this is just a basic breakdown, not all of them, but I tried to break some of the genes down into categories to emphasize that there are genes on the sex chromosomes that affect all functions of the body and not just reproduction. So when you go through some of these in terms of brain development, cancer, melanoma is a really interesting one. Prostate and testicular cancer, you wouldn't necessarily think that those would be on the X chromosome, even though they're more present in males. Cardiovascular disease, regulation of metabolism, apoptosis and glucagon storage, endocrine diseases, hematology, immunology, and this is really important now as we talk about the coronavirus and all the data regarding the sex differences in incidence and more mortality associated with that condition. When we think about the Y chromosome, there aren't as many genes on there, but there's important ones that direct the development of the gonads, and those are the sex determining factors. But there's also a group of genes on here that affect the autosomes, and those are becoming more important in our understanding of the development of coronary artery disease, immune signaling, and inflammation. So when you think about these genes related to coronary artery disease or immune function, and immunology on the X chromosome, you can really see that these would affect susceptibility to some diseases that are in regards to the immunocompetence response to immunization, inflammation, and so on that exacerbate, for example, cardiovascular disease. So keep in mind that these genes on the sex chromosomes do more than just regulate development of the sex organs. Every cell has a sex. But the genes on the sex chromosomes really do influence the development of the gonads, which produce the sex hormones. And there are two types of effects of the sex hormones. Activational effects, which are reversible. They're present when the hormones are there and absent when they're not. The best example that immediately comes to mind is a very visual component is that of pregnancy, that the hormones modify every system of a woman's body and enabling the fetus to be brought to term. Some of these changes are reversible once those hormones go away. Example, the expansion of the breast, the expansion of the cardiovascular system, the ability to volume regulation, and so on. The organizational effects, however, are non-reversible, and these would be directing the development of the sex organs. As the hormones decrease even with age, these organs may change their characteristics, but they do not go away. So hormones vary across the lifespan. They're present, both testosterone and estrogen are present in both males and females. But this is an important concept to consider when you're designing your experiments, because many times experiments are targeted for a specific age group of experimental animals, which may be reproductively competent or not. In aging, those hormones are going to be changing. With an experimental design for clinical trials, what is the age distribution of the individuals? And if you now go on to clinicaltrials.gov for the vaccine for COVID, you can see that there are many tests for dose response by age group, decade of life. And this is critical if we're going to be able to provide a vaccine, for example, that's going to be effective for the entire population. Here's this visual. There's the specific hormonal effects changing throughout life, especially those for younger children going into puberty, and then of course with pregnancy and the changes with old age, or aging, I should say. I don't consider myself old, but I consider myself aging. So just to reiterate that the sex steroids target every cell of the body as well. So we have this idea that people may not be interested in studying sex differences. I've heard this by several investigators when we run our workshops, similar to what we're going to be talking about today. They said, I'm not interested. I'm only interested in fundamental mechanisms. For example, I'm interested in the regulation of large conductance calcium activated potassium channels. And I'm sure many of you with, I don't know what all your backgrounds are, but you may be studying some regulatory system or cellular mechanism. But let's think about this for a minute, because just imagine that these large conductance calcium activated potassium channels may be present in both males and females. It's a fundamental mechanism, it's common and it's shared, but is it regulated the same? And that's what you have to ask. Is it going to be regulated the same in males and females? Because when you consider the activities of the sex hormones on the channel regulation, on enzyme regulation, these are non-genomic effects which have immediate activity, let alone the genomic effects which regulate genes transcription, which have longer lasting effects, not only on the development of or production of enzymes, but also in terms of their regulation and structural changes of the organs. So I think we have to keep in mind that we have these very dynamic systems where you have sex as a basic component of the nucleus and the genetic mechanism, but then layered on top of that are these hormonal interactions. So when we talk about cellular mechanisms, we talk about ion channels, biomarkers, stem cell, and transplant, but we have to consider that the mechanism and the outcome may be regulated differently in males and females in terms of coming up with a particular system or mechanism. And that's why it's critical to consider the sex of the experimental material, the age of the experimental material, and the hormonal status, because those three factors can't really be segregated. So this is just an example to show you how that regulation of a critical physiological parameter, cardiac output, might be regulated differently in males and females. And an example of this is that, you know, get to the same regulation of cardiac output, but during stress, women, females will increase their heart rate, while there's critical changes in peripheral resistance or a decrease in peripheral resistance in males, which is not often seen in females. So the outcome is the same, but you're getting to it due to two different mechanisms. And what would be the factors controlling the heart rate, what are the factors controlling peripheral resistance in terms of cardiovascular function, are really different between the two. And we need to understand those, given that cardiovascular disease remains the #1 killer of both men and women worldwide. So there's compensation. We have sex biasing factors. The chromosomes may reduce the effects of the hormones and vice versa. By eliminating one factor, we can uncover another, but you get to the same place in males and females by different mechanisms. And this is sort of the basis for thinking about or reframing your research proposal in terms of considering sex as a biological variable. Okay, so we've sort of reviewed some of the basic fundamentals of considering sex, the physiology of it, and what we want to now do is go to framing more of how you can consider some of these things in the design and development of your research grant. So to summarize, because sex matters in physiology and pathophysiology, because there are sex-specific hormones and chromosomes, and there are sex differences in the incidence, prevalence, etiology, symptomatology, response to treatment, morbidity and mortality, and health and disease. So we've talked about sex. Sex is biological. I want to talk a little bit about gender, because gender is a psychosocial construct which relates to cultural attitudes and behaviors. Now, there's a gender has usually been considered as divided into three realms: gender norms, those which concepts which influence the individual; gender identity, how a person identifies with the environment and the world and the culture; and then gender relations, how we interact with each other. Now, there are several attempts to measure gender scales. And I will propose that it doesn't really bring much value to applying a gender scale to understand the mechanisms or interventions that may be necessary to either improve health or treatment or outcomes. Because when we really consider what the components of gender might be, we can see we take down individual components which can be measured and identify where the intervention may be most important. You have to keep in mind that all these concepts of gender, lifestyle, diet, activity, use of recreational drugs, access to care, employment, what risks that may give to your health, how we interact with each other, how we interact with family relations, where we live. Do we live at altitude? Do we live at sea level? Do we live in a humid environment? Do we live in a dry environment? All these factors are going to affect how we interact with each other. But you can actually measure each one of these, okay? And then know where, for example, if there is a cultural component where women or men are maybe denied certain aspects of care or activity or interactions, that can be influenced. The same with employment. If you're exposed to environmental risk factors or pollutants, that can be addressed, that can be changed. Alternatively, with the biology, you can't really change your sex. Okay, that's chromosomally driven, and we're sort of stuck with those chromosomes in every cell. Your race is some of the characteristics are genetic. Age we can't control. We add a year every year, but we can modify how we might age, whether we remain active and healthy and eat right, and so on. Our hormonal status varies across the lifespan, and our reproductive history, another component of biology, which is very important, especially for women, because many adverse effects of pregnancy carry lifelong consequences to development of chronic diseases as women age. So when we're considering these two factors in biology, we use the words male and female. For gender, we use men and women. So the question then is, how can I include sex as a biological variable in my experimental design? So the expectations again are that sex as a biological variable will be factored into research designs, analysis, and reporting in vertebrate animals and human studies. Now this is really important: experimental design, analysis, and reporting. Now I would challenge many of you to go to your publications, which are high impact in your field, and pick up an article that you think is critical and see if actually the sex of the experimental material is reported. Especially in clinical studies, they may report the number of males and females, but that's the extent of it. There's no dichotomy of reporting the data or outcomes or baseline characteristics of that population by sex. Therefore, it limits the ability of that material to be used not only in designing future studies, understanding what parameters may influence sex as a biological variable, but also in meta-analyses. So let's talk about sex-specific conditions, because where possible, if you just study one sex, conditions related to reproductive or urological organs and processes are the key things that would define studying sex-specific conditions. We do that at our Specialized Center of Research Excellence at Mayo. We're focusing on conditions specific to women related to reproductive status, ovarian status, and use of menopausal hormones. That's a sex-specific condition. sex-specific conditions for men would relate to prostate and testicular diseases and conditions. Some established cell lines don't have sex identified, but you can identify the sex of the cells. And if they're established, sometimes the sex chromosomes disappear with continued culturing, and that's something that needs to be paid attention to. Data are needed from cell lines of other sexes too, so keep in mind that development of freshly harvested tissues is important. One of the things in cell culture you have to keep in mind, every cell has a sex, so if the cells are derived from a particular animal, know the sex of that animal. The media may contain hormones. For example, fetal calf syrup, you have to strip it over charcoal to eliminate those hormones, especially if you're interested in studying the effects of hormones on those cellular functions. And indicators such as phenol red may act as a estrogenic type material. It binds to estrogen receptors, which may modify your process. So media for your cell culture experiments is going to be an issue. You need to be aware of it. You may have cell viability may be an issue, but have to know that the hormones in that may be necessary for cell viability, but just keep it in mind that it's a variable that you can't control. So how do we really address sex and gender in a grant application? Well, I think this is critical and requires us to really think about our research question in a new way. Is there a sex-skewed distribution or prevalence of the disease? Is your hypothesis sex-specific? You can include men and women, and males and females in pilot studies and randomize the randomization of treatment. Is it sex-specific or gender-related recruitment strategies, depending upon if it's a human study or not? These strategies may be important in order for you to include enough of each sex to have statistical power to evaluate a sex difference, if that's your goal. And then data analysis is critical that you report sex and gender components and measures, consider effect size, and really in the data interpretation, talk about whether it's sex influence or perhaps gender-related trends. So we've developed a series of questions that can help you frame your research proposal in this new way. First of all, is there evidence of a sex difference in the condition that's being investigated? If so, what is it? This can be put in the initial paragraph or framing of your research proposal. Keep in mind, if it's not been reported, it doesn't mean that it doesn't exist. It may not be studied. And you can fill that gap if that's one of the things that you've discovered. It's not known. That's an actual purpose that can be incorporated into your grant application. Is there an experimental model of the condition or disease? There's animal models of all kinds of conditions out there, some of them mostly male animals. Does that really represent what you're interested in? And is it typical of the general population of humans? And also, does the experimental model demonstrate the sex difference observed in humans? This is really critical when individuals are using transgenic animals or mice for their experiment, that sometimes the sex of those transgenic animals isn't reported. And in the phenotype of interest may only appear in one sex compared to the other. So those are critical components that you need to investigate, especially in your experimental design, that investigators or review panels are going to be looking for. And finally, are data lacking from an experimental model in one sex compared to another? And if this is true, there are ways to use the data that's available in one sex to design experiments to include the other, which will also help you to calculate an effect size or power calculation for the number of animals that you'll need. So I have a series of examples here, and our time is a bit limited. And if we were in a workshop, I would go through. Let's see, I have to stop sharing. Go back to the picture because I was told you need to see me occasionally. So I have three examples of how to incorporate sex in these different aspects. But because of time, I'm going to just sort of pass through those real quickly. And at the end, and I think there's time later for additional questions, we can go back to those. Those will be in my presentation. So if you go online, I think this is being recorded, you'll see them. But this is where I miss the interaction too. If I was there in front of you, we could have an open discussion from you of what your ideas might be when you see these paragraphs. And I think that's really critical because you can teach each other rather than just have me being a talking head telling you, here's what you think. I would really want to hear what you all think. But so let's just show them very quickly, and then we'll go on, and then we can come back to those so that they're in the recorded portion. So here's an example from a human research study. I'll just leave it up briefly for you to read. Here's an example from an animal study. And here's one from a cell culture study. Now, I'm hoping as you look at some of these that you can look at your own research and develop your own questions in terms of how sex might influence that design. So you have to keep in mind that considering sex as a biological variable and identifying a sex difference is not the same as investigating why there is a sex difference. So if this is really the question of why there is a sex difference, the design of the experiment is going to be a little bit different and needs to be powered. The number of animals need to be arranged a little bit differently than if you're just observing that there is a sex difference or considering it as an interactive component of your outcome. Is there a sex difference? Then you consider the mechanisms or proposals. There may not be. That's okay. If there is, here's a schematic to help you understand or get to the whether it's the role of sex hormones, differentiating effect of the gonadal hormones. You can test for a chromosome effect or consider an alternative experimental design. Do you lose the sex difference along the way, depending on whether you add hormones, take them away, and so on. So some of the critical information that's going to be necessary when you address sex as a biological variable in your grants or even in your papers. And we used to put this into the methods of all the papers. And years ago when I was starting graduate school, we were told that you put in enough of the information so somebody can reproduce your experiment, right? So we would always report the species and strain, sex, age, reproductive status, and sometimes you would say whether they were caged singly or in groups or whatever. You'd talk about the temperature, you'd talk about the food, you'd talk about the lifestyle or the life cycle. Some of this information is not available in methodology now because with the idea of impact factor, the methods are what got cut a lot of times or relegated to supplemental material, which people don't necessarily read. But these are critical information. When you look at clinical demographics, these are the same kind of parameters that need to be accounted for in as influencing the outcome and the design of the experiment. The race and ethnicity, sex, of course, and sometimes that may not be reported where gender might be. Most studies our experience is that sex and gender are pretty congruent for reporting for an individual. Age, reproductive status, reproductive history, and especially for women, now it's not important just to ask the number of pregnancies and number of live births, but to understand whether there have actually been adverse pregnancy effects such as preterm birth, spontaneous abortions, gestational diabetes, preeclampsia, or hypertension. Marital status and habitation is critical for understanding family dynamics and stress that an individual may be exposed to, whether they're in an urban, rural environment, altitude, sea level. All those things are going to affect the physiological adaptation. Income and education are usually adjusted for in experiments to keep or to have that information available. So what about the reporting as well as the analysis? Well, this is critical to experimental design. And here's just an example. If you aggregate your data, and many times this happens with transgenic animals, you don't know what the sex is, you have a control and a treatment, or the genes knocked out, and here's the response, disease impact. But when you really sort this out by sex, you may see that the control and the treatment have completely opposite effects in the depending on sex. And of course, when you aggregate it, you're going to get the average of the biggest effect or no effect at all. So showing the data or dichotomizing the data by sex provides you additional information. But of course, as one of our former birth scholars would say, this is in the zone of no information at all, because you do not know the distribution of the responses within that. So there's now a movement within scientific journals to publish the individual data points to show those distributions. Visualization of the data matters. Here's, for an example, the using wild type or knockout animals, or males or females. It's pooled, you don't really see an effect, but you can get to this point with the knockout animals by a variety of distributions of sex within each categories. One where there is no sex difference, the red and blue dots showing the male and female animals, or there is a sex difference in one group, or the apparent difference, group difference, is really related to the fact that there is a sex difference in the response. So visualization of the data provides you really with insight as to what is going on with the biology or the mechanism behind it. I'll give you an example of this from our own work, where we looked at the presence of coronary calcification. This is a CT scan across the chest. This is the left coronary artery, and the white stuff is a big hunk of calcium sitting in there. Not a good thing. We looked at women who had normotensive pregnancies compared to those that had preeclamptic pregnancies, and if we differentiated between individuals that were hypertensive and non-hypertensive. That being said, if you looked at the aggregate data of the amount of coronary calcium between the women that had normal pregnancies and preeclamptic pregnancies, you see that there is a significant difference. But when we visualize the data by points, we see that there is a group of women that had preeclampsia that remained with no calcium in their coronary arteries. So although there's this difference, and we reported this difference, that having a preeclamptic pregnancy puts you at risk for the development of coronary calcification as you age, what about these women that don't have it? Seeing these data and the responses like this really informs you of, here's another interesting question. What's the difference between the women in this group that may protect some and put others at risk? And so visualizing your data has a lot to do not only with informing the scientific community, but really spurring new questions that may come up along the way. So when you consider sex in a statistical approach, really depends on what's your question. Are you understanding, is the main focus to understand the sex difference? Are you just studying a sex-specific condition? If there's a known sex difference, how well known are the sex differences in your disease in focus. And really, money is not a reason to not include both males and females, or to, and that was always an issue when this concept first came out. Well, it costs too much money to do males and females. That reason does not fly. So you'll read a lot of statistical sections of paper where they adjust for sex. And I would like to leave you with the concept and emphasize that adjusting for sex is not sufficient. Adjusting for sex is equivalent to assessing sex as a confounder, and it suggests that there's no interest in how the outcome of interest might differ by sex. So if you're adjusting by sex, that automatically says that you think there is a sex difference, and you're going to ignore it and look at the response independent of sex. But individuals or outcomes aren't independent of sex. So it doesn't tell you where that sex difference might be. Is it greater in one compared to the other than that difference is there? So it's not sufficient to just use it as an adjusting variable unless you actually know ahead of time what that difference might be. So you can stratify by sex, and this assumes that all covariants have the same effect. Okay, so it often looks like there's apparent sex differences which may not be statistically significant, and this may be due to a small sample size. An alternative way to look at your data would be to look for an interactive term, which is gives you more power to the number of to see differences if they are actually there. And if you use an interactive term first, then stratify, it actually brings you to better understanding. Now, when I presented this to some editors of scientific journals, one of the editors challenged me by saying that, well, not everybody knows statistics in order to do this. Okay, that's true. I'm not a statistician. The statistics I learned was Snedeker and Cochrane back in the day. That was the Bible of statistics. But we are now in an environment which encourages interdisciplinary teams. So there is no excuse for people at academic medical centers to not include a statistician on your team, or to gain expertise for some way. If you don't know the answer, as an investigative scientist, you ask the question. And then the other response was, well, it's sort of standard in the field, excuse me, for sex to be adjusted as, use it as a confounding element. And well, if it's standard in the field and it's not providing you with enough information, then let's move forward and change the way we do it. So for even journal editors to sort of dismiss it, that reporting data by sex is not interesting or critical, the idea this needs to go beyond just the basic scientists. And you all are in the position that you review papers, you can start thinking about it and your own data when you write it up in a new way. Okay, so again, I have a series of scenarios that the aims are to understand sex differences. If the aim is to understand the sex difference, make sure you have adequate number of both sexes. Then you need to consider if there is an uneven distribution of a condition by sex, you have a higher prevalence in one sex than the other. You really need to consider that in your recruitment and your feasibility and your power calculations. You need to be sufficiently powered to test for a sex difference. And those power calculations can be estimated by the interaction term. If your aim is not focusing on the others, sex difference, is there a sex difference known? And if yes, is your sample size representative of the sex difference? And you can incorporate sex in a statistical analysis, but not necessarily power for it. If no, make sure the sample size is representative of what is in the population, and state that there are no sex differences, but that's what you're going to examine. So let's look at a couple of examples of how this may play out. In your section of the grant, which requires you to address sex differences, we have previously reported that cross-sectionally men have higher levels of plasma total tau compared to women. However, studies on other plasma markers have not been assessed. Thus, we will assess sex difference in all analysis that include a sex interaction when needed. So they've looked at the literature, they understand what is there, what is needed, and how they're going to address it. Here's another example. Both males and females are included in the study. Severe traumatic brain injury is overrepresented in males, in which case our population is overrepresented as well. Sex is known to impact patient outcome, so we're going to control for it. And while we tend to control for this confounder in analysis, we will also conduct analysis for each sex independently. Okay, so you have a sex bias in the incidence. You know that the outcomes are influenced by sex. You can account for it, but you're also going to look at it independently. And then a third example, sex differences in a number of microglia and levels of cytokines are in the preoptic and parietal areas and so on have been documented. Although we have not observed sex differences in cerebral spinal fluid of immune markers in this one particular trial, we will continue to assess whether they exist in the proposed analysis. Okay, so if it hasn't been documented and you haven't found any, that's fine. We need to know that information as well too, but that doesn't mean you stop there. It means you continue to look as you go forward. This is what you were typically given when you were on a study section in terms of how to consider sex as a biological variable in the grants that you are reviewing. So every reviewer gets this in their packet of material when the grants arrive in your link, and it provides you what kind of consideration needs to be acknowledged in terms of the strength of the justification of the studies and the whether it's considered a major weakness or a strength. Now, I want to sort of give you some knee-jerk critiques that I have encountered either being on study section or read in comments for grants either that I've received or that some of our trainees have received. There is no mention of sex distribution of the topic under investigation. Well, duh, if you have to account for sex as a biological variable, mention it. Think holistically in terms of it. So just sort of dismiss this, just address it straightforward. It is unclear who compromises the patient or animal population under study. Well, again, let's not let the reviewers guess. Give them description of age, sex, and hormonal status. This is like fundamental basic information. But can you imagine it was unclear? I mean, really? How can you evaluate that proposal? No mention is given of how recruitment will be balanced by sex. Well, you know, if you're recruiting for a certain condition, where are you going to recruit? As they come into the emergency room and the physician's office, or you're going out into the community? Where do individuals go if there's a difference in terms of frequency to a gym or to a hair salon or a barber shop or a dental office or some activity? Do you have to have recruiting materials specific to sex? And if there's no mention, tell them what you're going to do. The study is underpowered to identify sex differences. If it's not designed to examine sex difference, you can also always suggest a secondary analysis to inform future hypotheses. And there's no attention given in the statistical section as to how the data will be analyzed. Well, see above. Report the data by sex and discuss whether or not, by showing the data points, you may not have enough, but it may inform you as to what you need to do to design your next experiment. So I gave you a lot of information here about, you know, again, I feel like I'm sort of a talking head and not really getting much interaction from you. You say, okay, you have all this, Virginia, and you're just going to leave us cold. No, I don't want to leave you cold. I want you to be able to have some tools at hand that you can refer to. There's some examples here in my presentation, but there's also a lot of other information available elsewhere that can help you. And one of those is a really nice little training module that was developed by the Canadian Institute of Health Research. This is a free module. There's 3 of them, and it talks about inclusion, just some basic concepts, which I've gone over today. There's one on animal experiments, and there's one on clinical studies. You can register for this. It's free. When you're done, you get a little certificate if you answer all the test questions right. I've never really passed the human one because I'm not all that great on some of the statistical analysis and questions. But the good thing about this is that they give you actually grant sections and ask you to rewrite them or to critique them as to whether or not sex is mentioned sufficiently. So I think it's a really good training module. You can do each one of them at separate times. Each one takes maybe less than 30 minutes. So it's a good introduction for all kinds of material. And we usually recommend the individuals attending our workshop go on to this website before they come to class. Of course, none of them do, but we still recommend it. Okay. In terms of interactive line plots and data presentation, there's Tracy Weisgerber was one of our former BERT scholars. They have these online programs. These are again free. Some of them are actually these interactive ones. You can actually see the drawings happen on screen. Again, these presentations, I've seen a lot of journals now requiring the visualization of individual data points. So those are good tools for you to use. So when we consider sex as a biological variable, I wanted to go over today, I wanted to make sure that we reviewed what the NIH policy is, and it's to consider sex as a biological variable in design, analysis, and reporting. We talked about the difference between sex and gender. Sex is biology, and an easy way to remember this, gender is everything else. It influences the biology, but we really need to dissect the components of gender, understand how it influences biology. What are the key components of the research strategy? Well, you have to have a question that considers it, starting with what you're going to think about your question in the terms of sex as a biological variable. What materials are you going to use? What tests are you going to run or interventions? How are you going to analyze the data? And of course, now in the reporting. And in a recent review of some of the papers, journals using sex, using cell culture from cardiac myocytes and cardiovascular tissue, we just recently published in the FASEP journal. We compared the results from 2012 to 2019. Many of the journals, we see an increase in the fact that sex of the cells is reported. That's a good thing. Some of them haven't changed. Some of them have actually gone down. So, you know, you're responsible for this. What are the responses to the knee-jerk critiques? Well, you have to anticipate what those questions might be and make sure the research plan addresses it. And one of a really easy way, but sometimes hard way to do this is to have your grant reviewed by a group of colleagues, people you trust. It's always, and the people that you know will give you honest feedback and actually read the application. I've had people, I've asked colleagues to read things and it comes back with it's okay. And I give it, and you know, that's not what you want to hear. You want to hear, what are your questions of this? If you were a reviewer what would the feedback be? So I'm going to leave you with that. And thank you for your attention. It's been challenging going back between trying to see you all and have you see me and deliver the material, which is very different than you don't see my animation when I get really excited about a topic or a particular point. But I think I've left a couple of minutes for questions. We could go back to some of those scenarios, or we can take questions from you all. I see there's chats going on here, but I can't keep up with them all. So I don't know, maybe a moderator could maybe look through those quick, or
So Dr. Miller, thank you so much again for coming and presenting to us. You do have one question right now.
okay.
The question is, do you have suggestions for inclusive gender categories to include when performing studies?
Well, you know, gender is really tricky these days. And so I know one of the good things about the new electronic medical record is that they have categories for sex and they have categories for gender. And they have categories for a lot of other things. If you don't want to include it, there's categories for male and female, man and woman, intersex, transgender. I forget all the other ones. Those are for clinical medical records. And sometimes there's a big question of how you include some of these categories in clinical trials and analysis. For basic science studies, it's always critical to just talk about sex. And even for human studies, if you are looking at a biological outcome, whether that be, you know, healing, immune response, vasodilation, I don't know, muscle contraction, bone mineral density, these are biological parameters, okay? And when you're talking about a biological parameter as an outcome, sex is the right word to use to define those differences between your populations. For gender, I think of what the individual is self-report, but I would encourage everyone to consider those components of the intake form, which might really implicate the biology. So especially related to activity, diet, living conditions, stress, and so on. I'm not a big, as you can tell, I'm not a big fan of gender scales. You know, how male or female you might, or how much of a man or a woman you might feel in one day or another. I mean, it depends on who you're interacting with, what kind of characteristics you might want to display. And that's not going to, you know, when you go home and you're exposed to the same stress, you go through the same, don't sleep, you eat badly, all those things. Does it really matter what you're calling yourself? And my take is not really, because you need to address those things that are going to be affecting your physiological function. It didn't answer your question, but it gave you an opinion. Well, let me ask the audience there, and they can maybe put in. So many of you have all submitted grants, and you've gotten critiques back. How many? I can't see show of hands. I don't know whether we can maybe write a little comment real quick. How many of you have actually gotten back in your critique something about sex as a biological variable, whether it was addressed or not? I can see some of you. I don't know if you want to just maybe nod. Some of you have your visual things off. So I see Dr. Rush is shaking her head and some other people. Would any of you want to share what some of those comments might have been so that you can help inform others? Dr. Rush, do you want to comment on what some of yours might be?
Yeah, this is when it first was introduced, I think, to NIH, but I mean, our comment was that our experiments that weren't adequately powered. So, and we had kind of suggested that in the initial experiments, like in the pilot studies, sort of, that we would include both, and then we would go back to using males, kind of hand waving about the cost, like you indicated, is never a reason. So It is a reason, but you're not allowed to say it,
You're
right?
not allowed to say it, yeah.
So it's just that we didn't have enough power, and then we put the cost factor in there, and we got slapped for that. So I've gotten a little bit wiser, but I'm here to become even more wiser, hopefully. So
Yeah, yeah.
and
I
thank
think
you, thank you for talking to us too.
Yeah, it's a pleasure. Like I say, I really am sorry that I can't be there and visit with you all in person. There's somebody else, Dr. Weinkoff. Tiffany, you shook your head.
Yeah, I've just had the line that sex was efficiently addressed. So I get that line just to say like, hey, you talked about it, and I haven't gotten any critiques though yet.
Okay, and there was somebody else, Isabel Mozzie. I'm sorry, I'm mispronouncing your name.
I must have been just shaking my head because I haven't had critiques on that yet.
Okay.
But I think Lisa, were you saying that you had comments?
So for grants, I think like Tiffany, I've gotten comments that it's sufficient, but for a paper that I'm currently revising to resubmit to a journal, one of the comments was that it needed to, I hadn't addressed it and I needed to address it.
Dr. Miller, you do have another question whenever you're
Okay, let me just.
someone asks, how do we address sex variables if we're working with breast cancer when I guess he meant when the male breast cancer rate it's
It's so
really
low.
low.
Yeah, that's a really good question because there are conditions which affect both men and women, but there's predominance in one sex or the other. That should be addressed straight up to make sure that the review team knows that. And I think one of the, you're only limited by the amount of material that you can get, right? And if you're not seeing any male conditions with breast cancer, or you're collecting material, it's just impossible to study it. On the other hand, if you do have individuals with that condition, that you can collect some material, you can put in there that we will collect the material, we will do the experiment, but we will not, and we can report it as an individual response, but we will not have clearly statistical power to evaluate a clear sex difference. But you identify that you don't have enough material, but you will still do the experiment on it, and you'll accumulate these data over time that eventually you may have enough to make a definitive conclusion about whether the response is the same or different between the males and female tissue.
Okay, great. We also have another question. Someone said, I would like to hear more examples of approaching sex as a biological variable in psychosocial research that isn't looking at a biological outcome. For example, I do psychotherapy and implementation studies. We might be interested on how an intervention spreads through a system and or on provider behavior.
Oh, provider behavior.
Uh-huh.
Okay.
No, that was a lot too.
Yeah, not the patient behavior, but the provider behavior. That's really an interesting question because there has been research which shows differences in interaction if you have the sex match or mismatch between the provider and the patient, whether it's female to male or man to woman, woman to man, and so on and so on. And that kind of information has been published for responses in emergency rooms in terms of triage and interactions with individuals. So I think if you're going to be looking at those differences, you need to account for that, the pairing of the provider with the patient and the implementation. I've also known of studies that have been specifically targeted to providers and to address interactions that they're using in a population that may be a mixed sex population or predominantly men or women. And this is particularly used lots of times in VA studies with veterans And so you can design your study specifically to train your providers, and then you can develop some tests to see if they've actually implemented that in their patient contact or information. So there are data available for emergency rooms on how to design some of those studies, and also for within the VA literature on addressing trauma and outcomes. Does that help? I don't know who asked that, but that's what I would recommend, or that's all I know so far.
Thank you.
Yeah. You know, I think with some of the psychosocial things, and I guess I'm most familiar with post-traumatic stress disorders because my daughter does that, and you know, we've talked about her research quite a lot. And the response to stress in male and females is very different, and it affects the body differently in terms of sort of the stress response and the long-term consequences and the cognitive impact of some of those things, especially related to sleep and so on. So the idea that the interventions for the psychological or component of the condition really sometimes has a biological component to it that would show a sex difference.
Looks like we have a follow-up question from the breast cancer question that was asked previously. The question is, when studying breast cancer using an animal model, do we need to include models with the male breast cancer?
Well, it depends on how many samples you have, and I would say that that would be a ripe area for research because there's not a lot of research done on male breast cancers. So if you have access to a source, I would say definitely go for it. And you know, it's one of those, I guess, high risk, high impact kinds of proposals where you might be able to get some funding for an ART-21, given that the material isn't that available. But then you have to consider lots of times these tumor cells are put into immunodeficient animal or so on, or genetic animal, and you know what animal you're going to put it in. You're going to put it in a male animal, not a female, right? And a lot of times that's sort of forgotten when you do these experiments on these immunodeficient animals. Are you really matching by sex? And I think it's important that you do initially to look at the responses. And that goes for stem cell kinds of experiments and transplant experiments too, because in transplant and the early cell stem cell experiments, there wasn't a lot of attention to sex congruity or homogeneity in terms of the transplants or the study, the experimental models. And I think that that really needs to be addressed with stem cell therapy. And now most of that is stem cells from an individual autologous rather than heterogeneity or heterogeneous going between sexes or between individuals. Okay, related to the locomotor function of people with post-stroke associated with training in high intensity target. Yeah, I mean, that I think rehabilitation medicine really hasn't addressed whether there's sex differences in responses to some of these programs. And the answer is, I don't know. How does one of your first, considering changes in male and female to increase cardiac output in terms of these locomotor function, just think of some of the training paradigms that are used for sports and so on. And are they really sex specific? And do they need to be? And I think the question is, do they need to be? And if they do, let's get on it. And even training for sports, when you think about young girls having more risk for ACL injuries with soccer and so on, are there training paradigms to strengthen those muscles and stabilize that joint? Would those be different from male? Or even with rehabilitation from knee surgery or hip surgery in older individuals, is there a difference in terms of the mechanics that would influence that training? So I think it's a good question. I don't know the answer, and I think, you know, do these training paradigms differ with stroke recovery? I think it's a great question. I don't know the answer. I don't know if there's data out there, but I think it would really be worth investigating. Some of the CRE for conditional knockout estrogen sensitive, so studies using CRE can only use males. That's really an interesting question, because some of those studies that activate some of these genes actually use tamoxifen to stimulate some of the responses. And I've often questioned when I've redone these grants, or my colleagues, whether that tamoxifen, one, is it metabolized to the active tamoxifen, because in humans it has to be converted, right, to the active form, and does that act as some kind of an estrogen agonist? And they tell me it doesn't. They haven't showed me any data, so I don't know the answer to it. And whether you all have thought about it, clearly you have, Dr. Rush.
Yeah, we actually got that question on an application once on the summary statement, so I never could quite track it down, whether if you are using a CRE that's, you know, that binds tamoxifen, whether you can only use males. We had put that in the grant and someone questioned it, so I still don't know the answer. That's why I'm asking you.
I don't know it either. When, you know, and I know that tamoxifen has to be metabolized in humans to an active form, and I don't know how it's delivered in these animals, and whether it's the actual tamoxifen or is it metabolized, and the metabolite. I could never get a good answer for that.
Yeah, it's given IP, but I don't know either whether, you know, it's active or not. So I've never figured it out. I never figured out the answer to that question anyway.
Well, if there are no more questions, I really appreciate you all for taking time out to listen to me today. I hope some of the information is useful to you moving forward, and that some of the resources that I've suggested can actually be worthwhile for you to implement into your work going forward.